In the following 8 weeks, we'll talk about medical microbiology. This is the part of this course. For the Chinese student, we have separate 2 different courses. Medical microbiology is the subject exploring the microbial world and its interactions with humans. So it's a very special field of microbiology. We are not learning general microbiology, but we are also learning the interactions between organisms and humans, how they cause disease, and how our body responds to microorganisms. Like if you have some infection in your body, how your immune system fight against the pathogens, how the pathogens are get rid of from your body. So that's the major contents of medical microbiology. We spend a little bit of time to know each other. We are here, right? Zhengzhou City. Probably here, Zhengzhou City. So where are you from? Let me try. So most of you guys are from here, right? Iran, Bangladesh, yeah? India? No, not at all. Because at the very beginning in 2018, more than half of the students are from India. Pakistan? One? Okay. Afghanistan? No. Turkey? Anyone from Turkey? No. Here? Morocco? It's here, right? Or here? I don't know. Maybe other countries in from Africa? Yeah. No matter which country you are from, even including me, well, thousands of years ago, actually 70,000 years ago, we were from here. So that's the sweet home of all humans, right? You don't know that. 70,000 years ago, our ancestors came from here, get out of Africa, and spread all over the world. So, in some sense, everyone in the classroom here, we are kind of relatives, cousins. Because 70,000 years ago, scientists believe that data show that only 200 people came out of Africa. A very small group, 200 people, a very tiny group. So we are all connected, related to each other. We are relatives, no matter where you are from. So, yeah, Africa is our sweet home. Africa, yeah. Africa is our sweet home. Not a long time ago, 70,000 years. It's a very short time. 70,000 years. So why it's a very short time? Let me tell you the big history from the very beginning later, okay? Okay. Ladies, you want keep quiet. It's a nice kiss. Keep quiet, okay? Yeah, something you never heard of, right? Well, let's talk about the history from the very beginning. A little bit information about me. Some of you know my name because I led your lab section. My name is Zheng. I'm teaching you your microbiology. I'm the only teacher here for microbiology. I don't know if it's good or not. Gender? Okay. Age? Don't ask, okay? Little bit about my education background. Yeah, okay. Now, I do have a PhD degree, okay? I got my PhD degree in molecular and cellular biology from Stony Brook University. And I got my bachelor degree in bioscience and also business management from a university called USTC, University of Science and Technology of China. You never heard of this university, right? It's one of the top universities in China, way better than Zhengzhou University. Yeah, Zhengzhou University is here, USTC is here. Yeah, top 5, I think. More information. I consider myself a scientist because I'm doing research and I have my own lab and we have science projects. I'm liberal, also feminist. Have you heard of the saying that a real man is a feminist, right? I'm not related to any religion, so yeah. I respect anyone who believes in your own belief. That's okay. But I'm not related to any religion. And I'm open-minded. Feel free to talk. Feel free to contact me with anything. But keep that in mind. I'm open-minded and I don't believe in God. Okay? If you have any question related to microbiology, you can email me or talk to me on WeChat. Send me a message. I'll try my best to get back to your question as soon as possible. All right? I prefer email. Okay. All right. So that's me. Today we will talk about the introduction of medical microbiology. Well, I'm teaching the course in a different way than other teachers because when I introduce microbiology, I would introduce from the very beginning. What's the very beginning? The beginning of everything, beginning of time, beginning of space. So I'll introduce a big history of life, of everything. So yeah, before we talk about that, when we learn anything related to biology, microbiology, or medicine, I would tell you 2 important skills you want to keep in mind. The first one is that when we learn the subjects, no matter what subjects, we should, well, look backwards and forwards. So we should look through the history and also the future. So we should think about evolution all the time. Even microbiology, even like in the future you work on tumor, on cancer, you also want to keep that in mind. The tumor in your patient's body is evolving all the time. So that's evolution. The other thing we want to consider in mind is that we should look over the whole ecosystem. When you study some bacteria, you are not only study the bacteria itself, you want also study the surrounding factors like temperature, environment, other bacteria, if it's a pathogen, how it interacts with its host. So everything you consider together as a whole picture. So that's the interactions. So keep this in mind. This is a big famous saying that nothing in biology makes sense except in the light of evolution. If you have this idea, this concept in your mind, sometimes you can think about the problem in real depth. You can think about the real problem and solve the problem with the real method. Nothing in biology makes sense except in the light of evolution. By this guy, Dr. Dobzhansky. He was a Russian scientist, but he worked in America later. Okay, let's talk about the big history. The big history begins from the Big Bang. You know the Big Bang theory, the TV show. The Big Bang theory is a theory of physics. It says that everything, matters, energy, space, and time, were generated at exactly that time point, 13.5 billion years ago. And from the Big Bang, it formed everything, all the energy, all the matters formed at that exact moment. And that's the beginning of our universe. And after Big Bang, 300,000 years after that, atoms formed, and different atoms collide to each other to form molecules. When we say, well, atoms comes together to form molecules, what we are talking about? That, exactly, that's chemistry. So chemistry is the study of the bonds between different atoms, right? And long after that, what's the time point is the formation of our solar system. Anyone know that? When the solar system formed, you know that. The sun, the earth, everything. Yeah, when did it form? It's about 4.5 or 4.6 billion years ago. That's the formation of our solar system. And soon after that, our Earth formed. And not long after that, 3.8 billion years ago, life on Earth appeared. So we enter the era of biology. We can study life. But for sure, at this point, the very beginning form of life was quite different from the life on Earth right now. So probably we cannot imagine how they lived on Earth such a long time ago. Because at that time, they cannot utilize the sunlight, and they can only get energy from chemical reactions. So they must be single-celled prokaryotic, even without cellular structure, the early life. But that's the point life formed on our Earth. And after a long time of evolution, see here, billion, right? After a long time of evolution, 2.5 million years ago, humans appeared in Africa. Here, when I say humans, it means that there were a lot of different type of humans, different type of species. Okay, and different names. They have a family tree, but that's 2.5 million years ago. sounds still a long time ago, right? But the next time point I want to tell you is that 70,000 years ago. That's not long enough, right? Right? So it's just a blink of time. 70,000 years ago, our ancestors, Homo sapiens, left our sweet home, East Africa, and then we came to Asia, Europe, and then spread all over the world. It's a very short time ago. And it was believed that only 200 people came out of Africa at that time. So we can say that everyone in the world, we are cousins, we are relatives, right? We separated only recently. And about 30,000 years ago, Homo sapiens, we became the only human species on Earth, which means that before that, there are other human species like Neanderthals, like in Asia, there are other species like Denisovan, that's another species of human. But about 30,000 years ago, all of them died off, and we, Homo sapiens, became the only species on Earth. So yeah, everyone in the classroom, you look different. Your skin color might be dark or pale, but we belong to the same species. We are very close. And 12,000 years ago, Homo sapiens developed techniques and to grow crops in the field. So that's agricultural revolution. And because of agricultural revolution, people can settle down in small villages. They don't have to travel. They don't have to hunt every day. So that we have villages, then we have cities, then we have civilization. But only in recent 500 years, we enter the era of science. So we only in recent hundreds of years, only in recent 500 years, humans began to study and realize the world with logic and evidence. So that's the era of science. Very short time, right? 500 years. And yeah, in recent 500 years, we learned a lot. We learned a lot of knowledge, and we developed a lot of techniques, including like computer, electricity, everything invented in recent 500 years. So all this big history tells us that we humans are not special at all on the earth. We are just one species of life on the earth. And we humans share common things with all the other form of life on the earth. Like we share the same DNA coding mechanism with bacteria in the soil. And we even share some same sequence with bacteria, right? And if you blast your DNA sequence, you might find some fragments, very short fragments, same as in bacteria. And also that all life on the earth are connected to each other. Okay, we need to rely on other type of life to survive. We need to eat meat, crops, vegetables, fruits, and even for microorganisms. We might believe that, well, they're not good for us, but actually most of the microorganisms are good for our health, and only a very small portion of them are pathogens which can cause disease in our body. So that's the general idea we want to master today. Okay, so that's a big history. The 70,000 years is just very short time. Okay, microorganisms. we are learning. What are microorganisms? You can just understand from their name. First, they are micro, which means very small. And they are organisms. Organisms, you can understand like as a creature or beings, which are too small to be visible through our naked eyes. So to study microorganisms, we need to use modern equipment to see them, such as light microscope, or sometimes you need to rely on electron microscope to see them. Okay, you use light microscope in the lab, okay, but I'm afraid that some of you don't know how to use it, how to focus on your cell. You want to improve that. And electron microscope, we do have several devices in our school. So if you do research in future, we do have electron microscopes here. Okay, all right. So this is the scale from big to small. Here is centimeter. If you have a ruler with you, you can take it out. Centimeter here. And here is a millimeter. That's the smallest length on your ruler. So if something is smaller than millimeter, for us, it's really hard to imagine it, right? For example, if you divide millimeter into 1,000 parts, that's too small to imagine. But that's the size of bacteria. That's micrometer. Most of the bacteria are on the size of micrometer. It might be less than 1 micrometer wide and a couple of micrometer long, like for bacilli. For cocci, the diameter is usually around 1 micrometer, so it's very small. We need to use microscope or electron microscope to see bacteria. For even smaller particles, like viruses, they are on the level of nanometer, from 20 nanometer to 300 nanometer. So it's very small. And we need to use electron microscope to see them. So these guys are the major subjects we want to study this semester on medical microbiology. We'll focus on several important bacterial pathogens and several viral pathogens. Okay? All right. The characteristics of microbial world. Let's talk about the microorganisms. There are three major characteristics. The first one is wide distribution and also with high diversity. And next one is interaction. Again, we talk about interaction. Any microorganism is not by itself. It must interact with its surrounding to get nutrients and with connect to each other to other microorganisms to form a complex network. Okay, the first one is wide distribution. Can you imagine where you can find microorganisms? Anywhere. So on the surface of this table, you can only see some dust, but actually it's all covered with microorganisms, bacteria, fungi, spores, everything on it. And in the air, it's transparent, you can see it's clear, but there are cells floating around in the classroom, right? On your skin, it's clean, but it's covered with microorganisms. You have you shake hands with your friends, okay? You will exchange microbiota with your friend. That's okay, it's safe. Okay. So everywhere In the air, in soil, in the water on campus, even in our drinking water, there are microorganisms. In plants, on the surface of plants, and also in animals, and even, in humans. There is one thing you probably never thought about it. In almost every cell of your body, every single cell of your body, there is a bacterium in it. Probably more than one bacteria in it. Do you understand that? In our cell, we have a special organelle called mitochondria, right? You know that? Mitochondria, that's the power plant of our cell. Mitochondria actually was some bacteria long time ago, but during the whole evolution history, long time ago, the bacteria invade into another cell and finally survive inside the cell and become our organelle, the cellular organelle. So mitochondria is from a type of bacteria living in almost every cell in our body. So wide distribution, right? And in our body, there's another data I will show you. In our body, if the number of your body is 1, the number of bacteria in your body is, can you guess the number? 1 or 2? Any other answer? So the actual number probably around 10. 10. So 10 times more of your own cell. The bacterial number in your body is 10 times more than your own cell. So everybody here in the classroom, you are not a person. You are a tiny ecosystem, right? Yeah, full of bacteria. So when you walk, you are just a moving ecosystem. So this is the first one, wide distribution, and also diversity. We can find a lot of form of microorganisms. Like some of them are eukaryotic. Eukaryotic means they have real nucleus, such as fungi. Some are prokaryotic, like bacteria. Some are non-cellular. They don't have even cell structure, like this, a simple viral particle. They are microorganisms. Even though we can say that a virus is not a real form of life, because they cannot live by themselves. But they are infectious. They can replicate themselves in the whole cell. And even some molecules, protein molecules, the prions, they are non-microbiotic protein molecules. They can also cause infectious disease. Has anyone heard of prions? Do you know prions? They should know the disease called mad cow disease. No? The mad cow disease. You know that, right? And also the sheep scrapie disease. Yeah. And those are the disease caused by protein molecules, prions. And those diseases are also infectious. Because when this protein get into your body, it can modify your own protein to the wrong folding state, which can cause disease. Okay? So that's the diversity of microbes. We will have a break, then we'll continue our lecture. All right. So we are talking about the diversity of microorganisms. This is a picture of the phylogenetic classification of all organisms. on Earth, and we can roughly divide them into three domains. One domain is eubacteria, so that's the real bacteria. So that's this yellow part. Another domain is this archaea, this brown part, archaea. And the third domain is eukarya, this green part. This is the part with real nuclei. So this three-domain family tree of all the organisms on Earth. And microorganisms are only defined by their size. So actually, we can find microorganisms in all three domains. Eubacteria, all the bacteria belong to microorganisms. And also archaea, they are small bacteria-like single-cell organisms. But till now, we don't have any evidence that archaea can cause disease in humans. So we don't talk about archaea in medical microbiology. So because they don't cause disease. While in this part, eukaryotic part, some fungi, they can cause infection in humans, such as some single-cell fungi, like the Candida albicans, which is a single-cell fungi, yeast-like fungi. It can cause disease in human. In fact, the human skin or soft tissue, and some mold can also set up infection in human, like lung infection with mold, and that's also fungi. A lot of fungus pathogens discovered in hospitals. So first we want to introduce the eukaryotic microbes. Eukaryotic means that they have real nucleus, true nucleus here. The nucleus is enclosed in membrane structure so that the membrane can separate DNA inside the nucleus from the cytosol. So that's one characteristic. The other characteristic of eukaryotic cell is that they have a lot of organelles in it, in the cytosol, such as mitochondria, Golgi bodies, all those membrane structure organelles in the cytosol. And those cells divide by mitosis. And we'll learn some fungi if we have time. They can cause like superficial infection, like infection on the skin, or cutaneous infection, subcutaneous infection, even some systemic infection. In my lab, some undergraduate students are now working on animal model. They infect the mice with fungi by directly injecting the cell into the bloodstream so that they can set up the systemic infection. And after infection, we can recover, we can find the fungi cells from the liver and also the the kidney. So we can set up the animal model to study the infection. And prokaryotic microbes, those are the cell without nucleus. You can see here, the DNA is in the cytosol, okay, but it's not not enclosed in membrane structure. So no nuclear membrane, no nucleolus. But they retain the basic cellular structure, and they have all their metabolic enzymes. So they can live by themselves, okay? And also in the cell, you cannot find the organelles, membrane organelles, such as mitochondria, Golgi body, no such organelles. And in this part, we'll learn a lot of bacterial pathogens. If we have time, we will also learn actinomycetes and also mycoplasma, which can also cause disease in humans. Here are some examples of prokaryotic pathogens. And this one, Staphylococcus aureus. It's a cocci, which can cause skin infection. Has anyone got sty when you were little? You know sty? Yeah, like here, yeah. So that most of the sty are due to infection by Staphylococcus. And also Streptococcus pneumonia, this one. Streptococcus pneumonia can cause lung infection. Salmonella typhi. It can contaminate water or food. When we eat those contaminated food, we can get infected in the digestive tract. Those are typical bacterial pathogens we'll introduce in detail in future. Okay, the next type is non-cellular microbes. So that's the viruses. They don't have cellular structure, no cellular structure, but only simple particles. The major components of those particles, only protein and nucleic acid. They have a protein shell, we call it capsid, and they have their nucleic acid and their genome. This nucleic acid can be DNA or RNA, but it cannot be both. Okay? Do you know the nucleic acid of COVID-19 virus? RNA, right? Single cell RNA, and positive sense single cell RNA, right? You don't understand that, but we'll introduce that. That's COVID-19 virus. Okay, this is an example of viral disease. The first one is chickenpox. Anyone got chickenpox when you were little? Wow, so many. Everyone? We should have vaccine available in your country, right? No? No? I'm too old. I forgot whether I got it or not. But here in China, all the kids can get the chickenpox vaccine. Now you have it. When you were a child, you don't have it. Okay, get it. Okay, good news. You've got chickenpox before, so you will never get chickenpox again. You are immunized. Okay, that's good. The good side of the story. The bad news is that the virus is hiding in your body right now, in your nerve cells. It will live with you for decades. When you are getting old, or when your immune system cannot work well, they can cause infection from inside. They come from the neuron, go through the neuron fiber, and then infect the skin cell. So the second time it causes disease, it's not chickenpox anymore, but it's a zoster disease. And this zoster disease is much, much painful. Okay, so that's the bad side of the story. But the good thing is that right now we do have another vaccine to prevent zoster, or at least you can reduce the pain of zoster. It's available now, but it costs a lot. Here in Zhengzhou City, you need to receive 2 shots, and each shot costs 1,600 RMB. The zoster vaccine. So, and it's only available for people who are who are 50 years older. So you guys cannot receive the vaccine at this moment. Okay, another virus, polio virus. Probably for your generation, you never see some victim like this, right? For your generation. But for the older generation, like your parents or your grandparents' generation, there are some victims like this. In China, ever since the 1990s, we don't have any poliovirus infection from the environment, the wild type infection. But there are some vaccine associated infections. At this moment, all over the world, there are only 2 countries which there are wild type infections. One is Pakistan, the other one is Afghanistan. So WHO is trying hard to get rid of those viruses by vaccination, but it's not easy due to religion or cultural problem. And hopefully, in a short time, we can finally get rid of polio infection, so that after that we can stop polio vaccination all over the world. Okay, we learn a lot of microorganisms, different types, diversity everywhere. And you might think that, well, we are endangered. We are living in a world which is very dangerous. But actually, it's not. For most of the microorganisms, they are not causing disease. If we fill all the microorganisms in this pyramid, most of them at the bottom, okay, majority of them, they don't cause disease. They are just friends. They live peacefully with humans. But there, in the middle of the pyramid, very few number of microorganisms. Most of the time, they don't cause disease. But in some special conditions, for example, when your body is weak, when your immune system is not working well, they can, well, cause disease, cause infection. We call them conditional pathogens. Okay? So only at special conditions it can cause disease. Only a very tiny number of bacteria can cause disease. They are professional pathogens. When they enter our body through the proper entrance, with enough number, they can cause infection. And those are professional pathogens. And those guys are the pathogens we want to talk about in the following weeks, the important pathogens. Okay, here's the data. In our body, we have 10 to the 13th of our own cell, but the bacterial number is 10 to the 14th, 10 times more than your own cell. So, yeah. But most of them actually are working for our body, and they produce vitamins. They help us to digest food. So most of them are friends. So, and we have to use them. And scientists did some experiment that if they leave the animal, the mice, in the environment without any bacteria, the mice cannot develop well, especially for the immune system. The immune system cannot get mature. So without those bacteria, our immune system cannot develop. So this is very important. And even for the plant, okay? On the surface and in the root area, there are large amount of microbes. And those microbes, including fungi and bacteria, can help the plant grow healthy and help them to get nutrients. Yeah, we should first have some idea that microbes, for most of them, they are friends. They don't cause harm to our body. Only a very tiny of them are enemies and cause disease. Because we are professionals in medicine and you'll be doctors in future, you should keep that in mind. You don't tell your patient that all microbes are bad. So yeah, most of them are good. Okay. Okay. The rest of the time, we'd like to introduce some history. Right now, we for most of the people, we can live a relatively longer life. So that's the average lifespan. For the whole world, the number is 67.2 years. And in America, this number is 78.1 years. And in China, it's close to this number, 78 years. And someone told me just now that in Iran, it's 75 years. You know that. So it's much longer than before. And if we check the number in history, during the Bronze Age is about 3,000 to 4,000 years ago, people can only live average lifespan 26 years. And in the medieval Europe, 400 to 500 AD, the average lifespan was 30 years. Improved a little bit. Only by the early 20th century, 100 years ago, it was 50 to 64 years. The major reason for a long period of time, people cannot live a long lifespan, is because of different type of infectious disease. They don't have any cure. They don't have any real treatment to kill bacteria or kill viruses. They just rely on their own immune system to survive. So most of the people cannot survive and live a longer life. And a lot of people die when they are very, very young, like several years after they were born. So that's the major impact of infectious disease. Here are some examples of infectious disease. The first one is Black Death. You know that, right? Black Death or plague. It's an infectious disease caused by a bacteria, gram-negative bacteria called Yersinia pestis. Every time when there was a break, the Black Death can kill 1/3 to half of the whole population. There is an example here from years 1346 to 1350. In 4 years in Europe, over 50 million people died. That's about half of the whole population within 4 years. So it's a sad story. And this plague influenced the history of Europe in a lot of ways. First, because a lot of people died, so that a lot of land are not available. So the survivors will have more sources, right? So the economy can grow. So that's the good thing. And the other way is that at this time, people believe in God. Almost everyone believes in God. But after the Black Death, a lot of people might think about, well, I believe in God so much, but all my relatives died. But it's not useful to believe in God. So after that, a lot of people just give up their belief. So that's the influence of the society. So that's the Black Death, the first one. And also smallpox. You never see any victim like this of your generation, smallpox. Because all over the world, we eradicated smallpox in 1980s, early 1980s. So after that, your generation didn't receive smallpox vaccination because we don't have any virus in the world. There are some strains frozen in the lab, in a security lab, but not in the population. So you didn't receive the vaccine. We don't have this infection anymore. Okay, smallpox. I never see a child like this. Okay, the next virus is influenza. So that causes flu. Right now it's flu season. Flu season. Well, probably no one got flu vaccine here in the classroom. Has anyone got flu vaccine this year? No? This year? You got? I got. Anyone? Oh, good, good. That's good. I would suggest everyone to receive a flu vaccine every year, flu shot every year. Because if you get infected, it takes you a couple of weeks to recover, and it's really painful. Because last year, even I got a flu vaccine, I also got infected by the end of the year, and I got high fever, coughing for over 10 days. Flu vaccine, it can cause a lot of death every year, especially for the very young people and very old people. And people over 80 years old, they are very vulnerable to flu infection. And about 100 years ago, around 1980, there was a flu breakout, and it caused about 15 million people die. This number is not accurate. Some people believe that there might be 100 million people die in a couple of years due to this flu virus infection. Okay, influenza. The next one is HIV, the human immunodeficiency virus. This is a new virus actually in history. If you put it in a long history, it was discovered by early 1980s in developed countries in America and Europe. At the very beginning, scientists believed that, well, they could handle it. They could make some vaccine and handle it. But unfortunately, till now, after 40 years, we don't have any vaccine available to prevent HIV infection. Because HIV virus is mutating all the time. The antigen is changing all the time. So it's really hard to develop any vaccine to produce antibody to neutralize the virus. And we don't have any vaccine available. The good news is that we do have some medicine available which can inhibit the virus so that the HIV positive people can live a normal life, but they have to take the medicine all their life to inhibit the virus. Hopefully, in the future, we could develop any universal and long-lasting vaccine to prevent HIV. Another virus, you should be careful because in the future you will work in the hospital. It's the HBV, hepatitis B virus. This is more infectious than HIV. And if you check this map here in East and Southeast Asia, in Africa, South America is hot spots. So, because you are professions in medicine, you have more risk of exposure when you communicate with your patients, when you give surgery to your patients. So I would suggest you guys, before you go to the hospital, check your immune status and see, just to check whether you have the neutralization antibody. If you don't have the antibody, I would suggest you to receive the vaccine again to activate your immune system so that you are protected. Get it? Yeah, because in Western countries, if you go to work in the hospital, they would give you the vaccine for free. And everyone should receive the vaccine, unless you don't want it. But here in China, in the following years, when you go to the hospital, they probably will not have that policy for you. And you want to help yourself protect it. HBV, it's more infectious, okay? And we do have vaccine, but we don't have any medicine to cure the patients. Okay. Then we introduce a little bit about the history of medical microbiology. Medical microbiology, well, begins when you can study microorganisms. But before the invention of microscope, people didn't realize the existence of microorganisms because they cannot see it, even though they can utilize it, but they cannot see it. So this begins with the invention of this device, the very first microscope, like this. It's very simple, but it can amplify particles over 200 times so that you can see microorganisms like this, okay? It was invented by this guy, Anthony van Leeuwenhoek. See his clothes. It's fabulous, right? This is a rich guy, and he was a businessman. He sells cloth, silk, and clothes. So he had a lot of money. And the reason he invented this device is to check the quality of silk, the cloth. Yeah. So this device was first utilized to check the quality of the cloth, but it can be utilized for research. So that's the first device of microscope. He actually made the lens by hand. Another doctor we want to remember is Dr. Edward Jenner. I think when we learn immunology, we learn this, right? When you study immunology, did the teacher talk about him? He was an England doctor. Was an England doctor. At that time, there was outbreak of smallpox, but he noticed that the milk ladies, they might get infection by the cowpox on the hand, but they never get smallpox. So the cowpox can provide some cross protection. So based on this, he used the cowpox as a vaccine, okay, to prevent smallpox. And it worked. And with this method, we finally get rid of smallpox all over the world. Edward Jenner. Okay. Another big scientist is Dr. Louis Pasteur. Louis Pasteur was French, but interestingly, his major is in chemistry, so he was not a doctor at all. But he devoted his life working on microbiology. Louis Pasteur. Here are some contributions by Louis Pasteur. He designed scientific experiments to prove that fermentation and putrefaction are caused by the growth of microorganisms. Okay. And also, he disproved spontaneous generation. You know spontaneous generation? Spontaneous generation was a theory for a long time, for thousands of years, that in a proper condition, life can generate by itself. For example, if you have a pile of hay, the bugs can generate from the hay by itself. So that's spontaneous generation. But now we know this is not correct, because life can only generate from cell. Any cell should come from their parent cell. So that's cell theory. And Louis Pasteur disproved spontaneous generation. That's very important. And also he invented pasteurization method to kill bacteria in liquid. If you have some milk in the morning or yogurt in the morning, or after class you would have a glass of wine, and those liquids are pasteurized to kill bacteria and fungi in it, so that you can keep wine for a longer time. Okay. And also he worked on immunology and vaccine. He invented anthrax vaccine. And also he invented the rabies vaccine, which is a virus vaccine. And he established the institute after his name, the Pasteur Institute. Even now, the Pasteur Institute is one of the most famous institutes all over the world working on microbiology. Okay, Louis Pasteur. Okay, the next guy we want to introduce is Joseph Lister. Has anyone heard of him? He's also a doctor in England, and his contribution to science is that he was a pioneer of antiseptic surgery. He introduced the concept of antiseptic surgery in hospital. He used the carbolic acid to sterilize the surgical instruments and also the room and clean the air, so that he can lower the surgery fever a lot, so that patients after surgery can survive. So that's very important. And now in the hospital, we are practicing these methods every day. So we can avoid the infection. We can save the patients. The last one is Dr. Robert Koch. This is a German last name, Koch. He identified the specific causative agents of several diseases, such as tuberculosis, cholera, and anthrax. And in his lab, they invented the method of solid medium, so that they add agar to the medium to make solid medium, and they can culture bacteria on the surface. And this technique, we are using it every day in the lab. But this is a revolutionary method, because when you culture the bacteria on the surface, you can isolate and purify the bacteria. So you get pure culture. While for Louis Pasteur, he cultured bacteria in the broth. So usually it's a mixture. Robert Koch. And he tells us the postulates. So this is a standard procedure and method to identify the pathogen of certain disease. Okay, the first step is that the organism must always be present in every case of the disease. You find all the patients, and you should isolate the same organism, same bacteria from all the patients with the same disease. And second, the organism must be isolated from the host. So you isolate the organism from the host, continuing the disease. And the healthy person shouldn't have the organism. After the isolation, you can inoculate this organism into some animal. And you can, the organism should cause the same disease in the animal model. And after that, you should be able to isolate the same organism from the animal. So it's a standard procedure to identify pathogen of certain disease. Well, right now, when we check these points, it's not perfect. All the points are not perfect, but it's a standard procedure. It's a general method to determine some pathogen, some causative agent of certain disease. Okay. By the end of 19th century, scientists began to discover another group of pathogens, which are much smaller than bacteria, which can pass through the bacterial filter. So that's the viruses. And those are the two important scientists who discovered viruses. Okay. In 20th century, we have a lot of progress in microbiology. Like Dr. Alexander Fleming found the first antibiotic, penicillin. Penicillin saved a lot of lives during World War II. So that's the first antibiotic which can specifically kill bacteria. And after that, more and more antibiotics were discovered, such as streptomycin and other antibiotics. But the problem is, well, we think that we can solve all the infection with antibiotics, but utilization of antibiotics causes another problem. So that's the antimicrobial resistance. Because when you use antibiotic, you give the microorganism some selection pressure. So the susceptible cells will die, but the resistant cells will survive. So finally, you will get more and more resistant cells, more and more resistant bacteria. So that's not good, because currently it seems that the antibiotics are not good enough to kill those resistant strains. This problem actually was noticed by Dr. Fleming at the very beginning. Okay, this is the note he mentioned when he got the Nobel Prize. He told people that I would like to sound one note of warning. It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And the same thing has occasionally happened in the body. So the person who discovered antibiotics warns people that you should use antibiotics properly, or you will have more and more resistant strains. So that's the problem we are facing now. So how to deal with this problem? Can we find more antibiotics? Yes, we can, but it's really hard. And in my lab, we are trying to culture microorganisms from the environment so that we can isolate new molecules, which might be able to develop into new antibiotics. We collect the water from the lake on campus, and during the summer break, we went to the Yellow River We try to culture bacteria from the Yellow River, and hopefully we can find something new, something novel, something better than the current antibiotics. But it's a lot of work. My students isolate over 2,000 colonies, and we have to test each colony separately. So it's a lot of work. Okay, some diseases have no effective method to control until now. We should keep this in mind. Even though we have modern science, modern technology, and hospital, but for some disease, especially infectious disease, we don't have anything to use. And some emerging and re-emerging infectious diseases still endanger the human. For a long time, people believe that infectious disease is not a problem anymore because we have antibiotics. But that's not true. We have emerging and re-emerging infectious disease we cannot deal with. Everyone here, we experienced the pandemic for 3 or 4 years, COVID-19. It's just a single cell, it's a single virus, a very tiny particle carrying only 20 to 30 genes, but it killed millions of people all over the world. And it took us a long time to develop the specific medicine and vaccine to prevent the COVID-19 virus. And what will happen in future? We don't know. Maybe next year there might be another new virus. Well, it's hard to say, right? Even the known virus, like flu virus, maybe some mutation can cause them very deadly. So it's hard to say. So we and future generations should continue to discover new microbes, new disease, and new methods of therapy. And still a lot of work to do. It's not a shame for you guys if you want to work on microbiology in future, okay? Okay, that's all for today.